Welcome to another Royal Astronomical Society's online public talk. Hello, I am Lucinda Offer, Education Outreach and Bicentenary Events Officer for our 200th anniversary of the Royal Astronomical Society. Our organization advocates for astronomy, geophysics, and space science in the UK since 1820. I'm excited to introduce to you today our bicentenary talk guest speaker, Rosie Cane, who will speak with us about microbes and how we can use bacteria for the search for life beyond Earth. Rosie Cane is a final year PhD student at the University of Edinburgh and UK Centre for Astrobiology. She completed her undergraduate degree in observational astronomy at the University of South Wales, where she specialized her final year dissertation on the use of terrestrial extremophilic organisms as an analog for potential life elsewhere in the solar system. Her work focuses on bacterial responses to multiple extreme environments and understanding the biochemistry of life. She also has a passion for inclusivity in STEM subjects, outreach and public engagement, and promoting physics and astronomy to GCSE level females. This is all extremely interesting, especially with 3 launches scheduled to start next week for missions to Mars, 2 of which will be looking for life and another studying the atmosphere of Mars. Today's talk may give us a better understanding of what the scientists operating those rovers remotely will be looking for. A quick reminder that there will be a Q&A after the talk, so please get your questions ready. There are places to post your questions for our speaker if you've joined us on Zoom in the Q&A. There's a Q&A box, and on YouTube in the chat box. So thank you, Rosie Cane, for being with us today and to celebrate with us our 200 years of the Royal Astronomical Society. On to you.
Great, thanks, Lucinda. And a huge thanks to the Royal Astronomical Society for inviting me to give this talk, and a very happy bicentenary to them. So good afternoon, everyone. My name's Rosie. I'm a final year PhD student at the University of Edinburgh, as Lucinda just said, researching astrobiology. And today I'm going to give you a brief and hopefully interesting introduction to how we can use bacteria in the search for life beyond Earth. Now, just for background again, I am an astronomer by undergraduate degree and have trained in microbiology over my PhD. So hopefully there'll be a good mix of both biology and astronomy within this talk. So with that, let's begin the virtual seminar. So in this talk, I will cover a brief introduction to astrobiology, a summary of what is life, what extreme environments are, how they can help us assess Martian and icy moon habitability, and finally, how we're searching for life beyond our own solar system via exoplanets. And a good way to begin this talk would be to define the term astrobiology. NASA defines astrobiology as the origin, evolution, distribution, and future of life in the universe. And it's an extremely multidisciplinary subject involving astronomers, biologists, chemists, geologists, physicists, engineers, so many different fields all coming together to help us to understand the conditions of other planetary bodies and their potential for life. Now, our planet, let me just move this. Our planet orbits just one star of about 200 billion stars in our galaxy, in a universe of maybe 150 billion galaxies. And with a universe so vast, astrobiology also seeks to answer the age-old question: are we alone? Are we alone in the universe? And this question can be exciting at the possibility of discovering new life, but equally terrifying. In the search for life in our weird and wonderful universe, we need to know what to classify as alive. Many organisms beyond our planet, and indeed some found in extreme environments of our own planet, will seem, for lack of a better term, very alien to us. But from our understanding of life here on Earth, we are able to come up with some key factors that help us define what life is. And defining life is not an easy task, but generally in astrobiology, we tend to go for the Darwinian definition of life, as supported by NASA, which states that life is a self-sustaining chemical system capable of undergoing Darwinian evolution. Now, Darwinian evolution basically means that all species of organisms arise and develop through natural selection. They do this using small inherited variations that increase the individual's ability to compete, survive, and reproduce, as introduced by Charles Darwin. And going by this definition, it's immediately evident that by studying life which is capable of surviving comparable environments here on Earth, it will allow us a certain level of predictive power when discussing the possibility of life on other planets. And in addition to a definition of life, we also need to establish what life on Earth has in common. All life on Earth is made up of what we call CHNOPS elements, which is carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, with carbon being the backbone of most biological molecules. Life also requires energy to grow, is able to adapt to its environment, can reproduce, can move, and can respire. And life on Earth is split into 3 domains, which you can see in the image on this slide. This shows the relationship between the domains of life, rooting from the first primordial form or last universal common ancestor. And we've got split into 3 main groups. We have archaea, we have bacteria, and this huge section up here, which is eukarya, which includes humans, which I've kind of blocked with my face, but we have a human there. And eukaryotes, you can see, are split into all of these different groups from plants, animals, fungi, and protists. And as we are looking at microorganisms in this talk, the main ones that we will focus on are archaea, bacteria, and these 2 groups of eukarya, protists and fungi. And just for a little bit more detail about some of these microorganisms. These are some of the microorganisms that we find on Earth. Generally, the main ones that we find are bacteria, viruses, algae, fungi, protozoa, and archaea. And some, like algae, can ruin a fish tank, others, like fungi, being great on pizza, and others which can send the entire world into quarantine. Now, all of these organisms are microscopic. We need a microscope to be able to see them, with the biggest microorganism being between 0.1 and 0.3 millimeters, which is still pretty small. Microorganisms are unicellular, which means they're made up of 1 cell, with the exception of viruses, which are not actually made out of cells, but made from single particles called virions. Now, most commonly in astrobiology, we tend to look at the groups bacteria and archaea, as these are found in some of the harshest environments on Earth, known as extreme environments. And these extreme environments are too harsh for humans to survive in. So why do we use bacteria in the search for life beyond Earth? Well, microorganisms are some of the most hardy and, importantly, most adaptive organisms found on the Earth. Due to their simplicity and rapid rate of reproduction, forming millions of cells in a very short time, this allows for a large amount of mutation and adaptation, leading to microorganisms surviving and even thriving in some of the most extreme environments found on planet Earth. These environments are teeming with microbes that can survive the harsh environments. The organisms that inhabit extreme environments tend to be called extremophiles or extreme tolerant organisms. Extremophiles come from the Greek word philos, or love, which means they are extreme loving, and not only grow under extreme environments, but they thrive within them and actually require some specific stresses like pH, temperature, and salt to grow. Extreme tolerant organisms don't actually require these specific extremes to grow, but can tolerate them, so are able to grow in these extreme environments. And archaea is one of the main groups to thrive within extreme environments. Although members of this group are generally less versatile than bacteria and eukaryotes, they're quite skilled at adapting to different environments, and they hold most records for the most extremophilic organisms. Some archaea are among the most extremophilic organisms known. For example, this is an archaeal organism known as Methanopyrus candleri, strain 116, which grows at temperatures of 122 degrees Celsius. That's the highest recorded temperature for any kind of life we've found on the Earth. Whilst we have the organism from the genus Picrophilus here, which is the most acid tolerant organism, which is known to grow at pH of 0.06, which is extremely acidic. And one extra point when we're talking about life in extreme environments is that we always tend to focus at them from an anthropocentric point of view, from a human perspective. And if microbes could express their views, they would call us extreme, as they would not be able to survive in the conditions that humans do. For example, your home office environment currently. Now, extremophiles were first discovered in the 1960s within Octopus Springs in Yellowstone National Park, a series of alkaline hot springs. And an organism called Thermus aquaticus was found here that could withstand the pH and temperatures of the environment. And since then, we've discovered a lot more microbes in lots of crazy environments on the Earth. For example, sticking to Yellowstone National Park, this here is the beautifully coloured Grand Prismatic Springs, which are named after their rainbow colours. This is kind of the poster boy of astrobiology and extreme environments because of the stunning views. And you'll see that around this lake, there are different rings of different colours. And these are actually made by different pigmented temperature-loving microbes, all surrounding this lake in different layers. And you can also see just how large this environment is to scale for the boardwalk around the edge and these tiny people walking around it. So this is just one example of an extreme environment, but there are many more out there. For example, the depths of Earth's oceans or the interior of volcanoes. And many extreme stresses actually expose more than one stress at a time. These environments with more than one stress are called multiple extreme environments and are really important in assessing the habitability of an environment. How are they important? Well, let's take a look at relevance to Mars. Because very often, studies into extreme environments tend to focus on just one singular stress at a time. For example, the low temperatures on Mars, which go as low as minus 150 degrees Celsius. However, we don't know how the addition of more than one stress in an environment affects microbial growth. So mixing together those temperature stresses with high amounts of UV radiation we get on the Martian surface from the lack of a substantial atmosphere, and adding in toxic salts we find on the surface of Mars, which include perchlorate salts, which are highly oxidizing and even found in rocket fuel. So we don't know how adding all of these stresses together actually has an effect on how bacteria grow, or any kind of microbe. So to really be able to assess the habitability of an environment on Earth or beyond, we need to be able to understand life's ability to survive in multiple extreme environments. And multiple extremes are a cause close to my heart, as they have been the focus of my PhD. I've been looking at how combinations of extreme stresses affect how bacteria grow. And I won't go into too much detail about my PhD, as that's not the focus of this talk. But experiments were carried out in a lab looking at the habitability of aqueous environments with relevance to Mars and icy moons in our solar system. And these involved looking at combined stresses of pH, salinity, nutrient limitation, and temperature, and looking at how extremes interact in an environment so we can improve our assessments of the habitability of other planetary bodies in our solar system or beyond. So from looking at all of the stresses that we find on Mars, Mars doesn't appear to be a very friendly habitat to life due to all of these multiple stresses. But there is hope, as the subsurface of Mars appears to have more favourable conditions for life, as this is shielded from the harsh radiation of the surface, so may be more favourable. But why do we always focus on Mars? Well, Mars has been a major talking point since the 1800s. At that time, Italian astronomer Giovanni Schiaparelli, a director at the Brera Observatory in Milan, began mapping and naming areas of Mars. He observed areas that looked like channels, calling them canali. Now, this was misinterpreted over the years to be canals, indicating that some form of intelligent life on Mars had built such structures, like we have to build the structures here on Earth. This was further backed by an astronomer called Percival Lowell. He also believed that these lines on Mars were actually canals, and these were used by Martians to carry water from polar ice caps down to the equatorial regions. Now, sorry to disappoint there, but as Schiaparelli was well aware, this wasn't the case. The Mariner 4 spacecraft imaged Mars in 1965, showing no evidence of this intelligent life on the surface. And since we've actually discovered that these so-called canals are actually ancient riverbeds on Mars. So although no intelligent life, we still get an exciting finding from this, as it indicates that there was once stable liquid water on the surface of Mars. And to further explore Mars, as Lucinda mentioned, we have some upcoming missions as well. So 2 of the upcoming missions that we have coming to Mars, which will help us in the search for life beyond Earth, are the European Space Agency and Roscosmos joint ExoMars mission and the NASA Mars 2020 mission. Now, the ExoMars mission has sadly now been pushed back from this summer to 2022. But when this does launch, this will have the primary goal of being able to determine if there's ever been life on Mars and to be able to better understand the history of water on the planet. The ExoMars rover here, called Rosalind Franklin, includes a drill to be able to access the subsurface of Mars and be able to tell us more about the habitability of that environment. The NASA Mars 2020 mission, which is still currently scheduled for launch between the end of this month and August, has the Perseverance rover, which will seek signs of ancient life and collect rock and soil samples with the possible return to Earth. And these missions, amongst the others we have launching over the next year or so, are really exciting, but getting to space is expensive. Is there a way that we can study the potential of habitability on Mars from here on Earth? Well, yes, we can use analog environments that recreate some of the conditions we believe are present on Mars. And one of these environments is the Basque Lakes, a series of small pools filled with high concentrations of magnesium sulfate salts, known as brines. And this environment in British Columbia in Canada represents a good analogue to the Martian surface, as we know that these magnesium sulfate salts are present on the surface of Mars. And I was fortunate enough to be able to visit this site during my PhD. So I was able to go to these individual brine pools and actually took some samples of the water and sediment to analyse back in the lab in Edinburgh. And because it was a Mars analogue site, we also took our plushy Mars with us to visit. But to tie this back with the title of the talk, how can bacteria on Earth help us with the search for life on other planets or moons? Well, one way is by using these analogue environments to find bacteria that can survive the conditions we know of on Mars or icy moons. We can then try and recreate these conditions in a lab to see how the conditions can affect how or if bacteria grow. We have a multitude of these analogues on Earth, seen in this figure, which match to different parts of Mars, so we can explore how its geology may affect habitability. So, for example, one of these extreme environments we have is Iceland, which has highly volcanic regions which match to the volcanic tharsis regions on Mars. We have the Danakil Depression, which is geothermally heated to be one of the hottest environments on Earth during summer and could potentially be an analogue to past conditions on Mars. We have the Basque Lakes, which I previously mentioned. We also have Kangerlussuaq Field Site, which is an area of permafrost in Greenland, which can be used as an analogue to the poles of Mars. There are the Makgadi salt pans, which were once a really, really salty lake, where the water then evaporated and left only salt deposits on the surface, and the Rio Tinto river, which is red blood and kind of alien in colour. And this has some similar salts and minerals to those detected on Martian soils on the Martian surface. So all of these are very exciting environments and can hopefully tell us a bit more about the habitability of past or present Mars. And following this, what would life, any kind of potential life on Mars, have to survive? What stresses do we know are present? Well, we know that there are high levels of radiation because of the lack of a substantial atmosphere. There are also low temperatures. We have high pH soils. The Curiosity rover found 2008 alkaline soils on Mars, and high salt concentrations, again, including these toxic salts and these highly explosive perchlorates that are found in rocket fuel. But there are so many more places to look at the solar system than just Mars. So if we move on from Mars and further out into the solar system, there are several more places we think could be promising for life. And these include some of the icy moons of the gas giants. There is the moon of Saturn, Enceladus, and the moon of Jupiter, Europa, which are both icy moons thought to have liquid water oceans beneath their surface. And few worlds in our solar system are as compelling as Saturn's icy moon, Enceladus. A handful of worlds are thought to have liquid water oceans beneath their frozen shell, but Enceladus sprays its ocean out into space, where spacecraft can actually sample it. From these samples, we've actually determined that Enceladus has most of the chemical ingredients needed for life, and likely has hydrothermal vents spewing out hot mineral-rich water into its oceans. With a global ocean, unique chemistry and internal heat, Enceladus has a promising lead in our search for life on other worlds. And also in the search for life beyond Earth, we can't forget Jupiter's moon, Europa. Decades ago, science fiction offered a hypothetical scenario. What if alien life was thriving in an ocean beneath the icy surface of Jupiter's moon, Europa? The notion pulled Europa out of obscurity and into the limelight where it's remained, stoking the imagination of people both within and outside the science community who fantasized about humans discovering life beyond Earth. And this fantasy, however, may be grounded in reality. Ground-based telescopes on Earth, along with the Galileo spacecraft and space telescopes, have increased our confidence for a subsurface liquid water ocean on Europa. We think Europa's ice shell is 10 to 15 miles thick, floating on an ocean 40 to 100 miles deep. So while Europa is only 1/4 the diameter of the Earth, its ocean may actually contain twice as much as the water on Earth's oceans combined. This does raise a bit of a problem, however, as if we observe life here on Earth, it was long believed that the life cycle required sunlight in order to be sustained, a source of energy. With the depth of the ocean and the thickness of the ice covering Europa, there could be no chance of development of life, right? Well, in 1977, while exploring an oceanic spreading region near the Galapagos Islands, scientists discovered hydrothermal vents. To their amazement, they also found that these hydrothermal vents were surrounded by a large number of organisms that had never been seen before. And these organisms were drawing their energy from hydrogen sulfide pouring out of these vents. Following this, if a similar mechanism could exist at the bottom of Europa's ocean, then life could be a real possibility, surviving using similar mechanisms. And we can summarize that the main stresses encountered in icy moons would probably be the low temperatures, high levels of salt, capable of keeping these oceans liquid at such low temperatures, and high pressure from the thick ice layer above them. Now, as these moons are so far away, there are actually analog environments that we can use on Earth to study them. One of these is Lake Vostok, a subglacial lake in Antarctica, which represents a good analog site to the icy moons. Temperatures in this lake are approximately -2 degrees Celsius, but the lake remains liquid due to a 2-mile thick ice sheet above it, exerting high amounts of pressure and keeping the water liquid. If any life survives in this environment, it would have to depend on a different energy source from sunlight. It's believed that organisms may get their energy from the chemicals in rocks in the environment, or there may be hydrothermal systems present like those detected on the Galapagos Islands. But the best way to determine if there's life on Enceladus or Europa is really to go there. And there are currently no planned missions to Enceladus, but there are several upcoming missions to visit Europa. This includes the ESA JUICE mission, or Jupiter Icy Moon Explorer, which is due to launch in 2022 and will fly by Europa alongside other moons of Jupiter, Ganymede and Callisto. Following this, we also have the NASA Europa Clipper mission, which is due to launch in 2024 to investigate and confirm the surface conditions on Europa. Hopefully, this will be followed by a Europa lander, which is not a confirmed project yet by NASA, but a lot of work is going into it. And this Europa lander would allow us to access Europa's surface and analyse the conditions to understand its potential for life. Now, there's another moon in the solar system that has potential for life as well. And this one again belongs to Saturn. And this is the moon Titan, which is an extraordinary and exceptional world. Among our solar system's more than 150 known moons, Titan is the only one with a substantial atmosphere. And of all places in the solar system, Titan is the only place besides Earth known to have liquids in the forms of rivers, lakes, and seas on its surface. Now, although this might sound promising at first for life, these rivers, lakes, and seas are actually made up of liquid hydrocarbons like methane and ethane. We do think, however, Titan may have a subsurface water ocean like Europa and Enceladus. So perhaps Titan's subsurface ocean could be a place to harbour life as we know it. Whilst its surface, lakes, and seas of liquid hydrocarbons could conceivably harbour life that uses different chemistry to what we're used to. That is, life as we don't know it yet. Also, Titan could just end up to be a lifeless world, but where's the excitement in that? We like to remain optimistic in astrobiology and searching for life beyond the Earth. So let's think about what kind of life would be able to survive on Titan. Well, we know that there's a large amount of methane on Titan, and sadly, these creatures aren't the culprits as they are for a majority of the methane found on Earth. We think that maybe life similar to methanogens on Earth, which are archaea that produce methane as a part of anaerobic respiration, could plausibly survive on Titan. Although living things on Earth, including methanogens, use liquid water as a solvent for their chemical reactions, it's conceivable that life on Titan might instead use a liquid hydrocarbon such as methane or ethane. The fact that Titan appears to have these lakes of liquid ethane or methane on its surface, as well as the rivers and seas, suggests it could support a hypothetical non-water-based life. It's been speculated that life could exist in the liquid methane and ethane that form the rivers and lakes on the surface, just as organisms live in water on Earth. Such hypothetical creatures could take in hydrogen in place of oxygen. They would then react it with acetylene instead of glucose and produce methane instead of carbon dioxide. By comparison, some methanogens on Earth obtain energy by reacting hydrogen with carbon dioxide and producing methane and water. But to find out more about the habitability of Titan, we have the Dragonfly mission, which will sample and examine sites around Saturn's icy moon. This is a NASA mission and it will launch in 2026, arriving at Titan around 2034. The rotorcraft, designed to look like a dragonfly, will fly to dozens of promising locations on Titan, looking for prebiotic chemical processes common on both Titan and Earth. And Dragonfly will mark the first time that NASA has flown a multi-rotor vehicle for science on another planet. And as you can see in the images, it's got 8 rotors and it flies a bit like a drone. Now, Titan can also act as an analogue to the very early Earth and provide clues about how life may have arisen on our own planet. During its mission, Dragonfly will explore diverse environments from organic dunes to the floor of packed craters, where liquid water, complex organic materials key to life, may have once existed together for tens of thousands of years. Its instruments will study how far prebiotic chemistry may have progressed, and will also investigate the moon's atmospheric and surface properties, and its subsurface ocean and liquid reservoirs. Additionally, and importantly, the instruments will also search for chemical evidence of past or extant life. And now, finally, to zoom out beyond our solar system, take a look at exoplanets, planets that orbit stars other than our sun. And the James Webb Space Telescope, now due to launch in March of 2021, will be able to tell us more about the potentially habitable worlds outside of our solar system. Webb will tell us more about the atmosphere of extrasolar planets, perhaps even finding the building blocks of life elsewhere in our universe. And it will do this using spectroscopy. Now, spectroscopy is simply the science of measuring the intensity of light at different wavelengths. The graphical representations of these measurements are called spectra, and they're the key to unlocking the composition of exoplanet atmospheres. When a planet passes in front of a star, the starlight passes through the planet's atmosphere. If, for example, the planet had sodium in its atmosphere, the spectrum of the star, added to that of the planet, would have what we call an absorption line, in the place of the spectrum where we would expect to see sodium. This is because different elements and molecules absorb light at different characteristic energies. And this is how we know where in a spectrum we might expect to see the signature of sodium if it is present. And the same can be applied to other elements that we might be looking for. Now, in addition to looking at exoplanets, the James Webb Space Telescope will also analyse locations a bit closer to home, and it will actually investigate the plumes of Enceladus and Europa, giving us a bit more of an idea of the habitability of those icy worlds. But the ultimate goal of the James Webb Space Telescope is to find a planet with a similar atmosphere to that of the Earth and detect elements indicative to life, such as these Chinox elements. So finally with that, I hope you enjoyed a quick introduction to how we can use microbes to search for life beyond the Earth. If you are interested in more astrobiology goodness, the Royal Astronomical Society Supermassive podcast actually has a new upload looking at how we are searching for alien life, featuring Professor Jane Hargreave from Cardiff University. And you can find that on their website if you'd like to hear more about astrobiology. But otherwise, thank you for listening, and I'm happy to answer any questions.
Brilliant talk, Rosie. Thank you so much. And we actually already have a request to make sure that this talk will be available online, which it will be, because we've been live streaming on YouTube. And so you are more than welcome to link to this talk later if you wanted to share it with Facebook. This request was to share it with their Astronomical Society. So lots of really great current information about searching for life in our solar system. Really cool. So we've got several questions already in the queue for you, Rosie, and I'm going to get to them, just giving some people some time to maybe think about what they want to ask, and you can post it to the Zoom Q&A chat box if you haven't seen it. It's available to all our attendees. Also, if you're joining us on YouTube, you can also leave a question in the chat box there for us. So let's get to a few. Let's get to a few questions here. So we'll get started. All right, one of the questions from Andy asks, how can we find life based on a completely different system, such as like something that's not carbon?
That's a very good question. That's the main issue with this, is that we can only search for life that we know. So we could end up going to another planet or moon, and there could be something that just looks like a rock to us. But that could be a completely different form of life from what we know. It's one of our limitations. We can only search for life that we know what we're looking for. So it would be very, very difficult to search for another thing, but there are people working on this. There are definitely people looking at trying to model what different life would look like if it didn't have carbon as a backbone, if we put different things in there. So it's hopefully something we'll be able to do more in the future. But currently it's generally looking for life as we know it, and we know what kind of signatures we're looking for.
Okay, and good question. Another one, if astronauts come back from another planet or a moon of a planet, is there a chance that any organisms brought back could be more dangerous than COVID-19? So something that may affect humanity here on Earth.
Oh gosh, that's an interesting and topical one. I don't know the exact logistics of what happens when astronauts come back, but I imagine they might have to be sterilized, have everything that they were wearing taken off before they go carry on and basically interact with other people. So I imagine there are precautions taken just in case anything like that happened. So yeah, we don't really know of any viruses surviving. It's not my specialty. I look at bacteria and microbes. So as far as I know, not know any viruses that have been subjected to these extremes and have survived.
Yeah, it's been 50 years since we sent humans off Earth to another celestial body like the moon, and when they came back, they were in quarantine for a while. I would imagine things on the moon get sterilized pretty easily
Yeah.
with no atmosphere. But yeah, that's a really good question, and I guess it's up to that
Mhm.
space organization how they want to handle their program. And there is a lot about planetary protection that people have to think ethical questions that we have that have yet to be answered. So it was a really good question, something still
Yeah.
unknown about it. Okay, next question. Are you also simulating the Mars subsurface atmosphere in the lab analog experiment? Can you do an analog subsurface atmospheric experiment?
Um, it's not something that I'm looking into, but so we can recreate the main atmosphere. So we have a Mars simulation chamber at the University of Edinburgh, which essentially is a metal box that we can recreate the atmosphere, the temperatures and the pressures and the radiation of Mars. So within that chamber, it will be possible to put some kind of a simulated crust and look at the microorganisms underneath, which some of my colleagues have been looking at to see if anything can survive under that. So it would kind of be possible to recreate, but it's not something that I've worked on myself to be able to tell you more about, sorry.
Well, I have to say, I love the curiosity and the thinking, and there's still lots of science to do, which is, one of the things you advocate
Yeah,
for more STEM, pathways to a career. So maybe something they want to look into and research for themselves. We need more researchers.
yeah,
So absolutely.
definitely.
So this is a good one. Here we go. Does the meteorites which hits the Earth support microbes? Do they support microbes on them? Are there any evidence for such meteors or asteroids having microbes on them?
Ooh, that is a good question. Again, it's not anything that I've really worked upon. I would imagine that the impact with the high amounts of temperature or anything would destroy anything that was on that meteorite. But that doesn't mean that we can't analyze it further to see what kind of organics are inside it. So whether that is from a rock that could have had life, say if it was on Mars, because we can see what's inside it. So maybe it's got some of those Shinops elements or something that could support life. So it's a really interesting question. Yeah.
Yeah, I know there's so much, and there's only so much you can do and spend your time on. But I think some of the ideas have been like maybe some atmospheres are trapped in the meteorite somehow and can travel through space, and possibly when they land on another body, then and I don't know if that's how sometimes scientists can study other planets from these meteorites that we find on Earth. Also, there was like the Allen Hills, which I think it's always questioning yes or no whether whatever they found from that meteorite from Mars was a fossilized form of bacteria. So lots of things to still look into and study and know about. And hopefully, with one of these missions going to Mars, and like you mentioned, NASA's mission with Perseverance is going to be caching, and then we have to like really hope that those samples sometime in the future will be returned to us, and that's always another thing to wait on and find out whether that's going to happen or not. Otherwise they stay on Mars. But one thing that's kind of interesting, and I don't know if how much of our audience has been keeping up, or if you know this, I heard that to calibrate one of the instruments on the Perseverance rover, they're going to be using a Martian meteorite that fell to Earth, and they actually got the sample from the London Museum of Science. So it's coming from the UK. They've given NASA a sample of this Martian meteorite, and they're putting it on the rover as to calibrate one of the very sensitive instruments looking at biochemistry. So that's really kind of cool that it's got this story, and we have sent Martian meteorites back to Mars, but only on traveling to Mars. Maybe it stayed in orbit, I don't know. But this is the first Martian meteorite that will be landing back on the surface of Mars, being returned to home in a little way. Little anecdote there. Okay, next question. Any thoughts on the possibility of subsurface water on Pluto?
Ooh. It's not one that generally comes up when we talk about subsurface oceans. And quite often we think that the oceans are actually formed on planets like around planets like Jupiter and Saturn, because these planets are so big that they actually pull these moons towards it. And then the other side of the moons, there are lots and lots of other moons, up to 80 in the case of Jupiter and Saturn, that are also pulling these moons in other ways. And this is kind of melting the interior of the moon and causing the ocean. So it seems to me unlikely that anything like that would happen on Pluto, just because it doesn't have that same kind of setup, and it's it would be so cold for any of that to melt. But we're discovering more and more things about our outer solar system all of the time. As we manage to get more spacecraft out there, we're discovering more and more. So hopefully eventually we'll find out more about Pluto as well. But it's not something that I'd say would be that likely.
Okay, good question. Rita asks, any chances that there's a life totally different from the life on Earth that does not require the conditions we need for survival?
Yeah, good question. Again, it kind of comes down to we can only focus on the life that we know, and we only know the life that we've discovered here on Earth. But it wasn't until the 1960s that we found these extremophiles living in these environments on Earth that we thought nothing could survive in. So it's a really interesting question, and yes, it's very possible. Again, like these archaea, which are really tolerant to extreme environments. Like there's so many things that are unknowns, and yeah, it's all really exciting just trying to unravel this big question of, is there life out there, and if so, what kind of life it is and what it can survive.
Okay, great. I'm going to go to YouTube just for one question from Seraphin, who asks, missions to faraway planets and moons are very expensive, like you mentioned in your talk, which is absolutely true. Is the purpose of these missions purely to create an understanding of extreme environments, or could there be a commercial benefit as well? That might, what is your thoughts?
Yeah, there's definitely commercial benefits. For all the engineering that goes into making these spacecraft. There's always uses for the different things that are used in them. In the past, spacecraft that have had cameras on them to go to different moons have ended up being used for medical purposes. So a lot of the stuff that has been made for the use of spacecraft can be repurposed in other fields of STEM subjects. And it is telling us more about these extreme environments, but also just what there is out there, like what elements we can find, what kind of conditions we would expect to be there. So yeah, they can be reused, and it's quite good in that way that it's all quite multi-purpose and different science subjects can share technologies and use them between themselves.
Okay, we actually, our questions have increased, but we still have a good 15 minutes, so this is good. So we're going to keep you busy all the way up until 2 o'clock. I hope you don't mind. Let me know if you think that's right. Okay, so Rachel asks, she says, can extremophiles hibernate? And if so, how long they can remain in stasis before revived?
Yeah, definitely. The best kind of way to explain that is probably to use tardigrades as an example, which I'm sure a lot of you will have heard, especially on the news the last couple of years, with the mishap where a spacecraft of tardigrades crashed onto the lunar surface. So tardigrades actually, when they're in extreme environments, the way they survive is they kind of dry up and they go into what's called a tun state. And they essentially just go to sleep while all the stress is happening. And then as soon as they are rehydrated, as soon as they have liquid water on them, they can be absolutely fine again. So like they sleep through all the stressful environments and wake up when everything's normal again and they're fine. I think it's been over 100 years that a tardigrade has been in this state and been reanimated again. So it's really interesting how they can do that. And it's a really cool way of being able to survive these environments, and just a good way of dealing with stress in general.
Great. Okay, thank you for that. Just keeping track of all these questions coming in, and mentioning tardigrades on the moon. Susie's one of your attendees on the talk says she loves the tardigrade toy that you have. I see also, do you have tardigrade wallpaper? Oh, I'm really jealous. Is that what that is?
I do. Tardigrade fabric, yeah.
Oh, fantastic. Okay. So thank you for that, Susie. So Neil Barnes asks, have we taken extremophiles into space, such as the International Space Station, to see how they react to, say, higher radiation or high temperatures? And he wonders if epigenetic processes will train Earth-based bacteria for potential seeding on Mars, if Mars was found barren.
Yeah, yes, extremophiles have been sent to the International Space Station. There's actually been quite a few experiments that have come from my lab in Edinburgh for that. So one set of experiments was not extremophile sent up, but some microbes were taken from a cliff in Devon, and they were actually attached to the outside of the International Space Station just to see if anything could survive. And these organisms came back and they could survive, they could replicate, they were fine. And we've also recently had an experiment called BioRock, which one of my colleagues has been working on, which is looking at if you send bacteria to the International Space Station, can they biomine? Can they, if they have a rock near them, can they actually take the nutrients and elements out of that rock to be able to survive? And just looking at that. So there's lots of experiments like that going on as well. I know the International Space Station take on a lot of different astrobiology experiments. But I don't know about all of them, just the ones that have come out of our lab. And in terms of the second one, sorry, Lucinda, could you repeat the second part of the question?
Yeah, let's see. Oops. I have to find it. There's so many questions. Right, the second question was, and they wonder if epigenetic processes will train Earth-based bacteria for potential seeding on Mars if Mars was found to be barren.
Oh, that's not something I know the answer to, I'm afraid. Potentially, we'd have to think about what the process is and what the aims of that would be. But I'm unsure about that one, sorry.
Yeah, some people wonder, is there life on Mars, or will we bring Mars to life? It is a difficult question to answer. There's lots of ethical, issues around it, and planetary protection, as we mentioned before. It's a really great question and something that we still have to answer. Yeah. Just wanted to mention, Dudley has shared with us that the question about Pluto and subsurface water ocean harboring life was recently shown on a BBC program. And so I think that's why, yeah, so maybe something we need to watch. Yeah,
definitely. Thank you for the recommendation.
Yeah, so I don't always have the time, but definitely going to put it on my list when I next. Yeah. Okay, thanks for that.
Yeah, that sounds interesting. Thank you.
Yeah. So Stella asked the ultimate question here: when will Webb telescope fly? We're all waiting on the Webb.
Yeah, hopefully soon. Hopefully next year. I think it's March scheduled for, so fingers and toes crossed that it will launch in March, because it's going to give us some really exciting results. And yeah, we've been waiting for a while for them. I think people are starting to get quite impatient now. But obviously it's for a good cause. They're having to make tweaks to the telescope. It's going to make it more robust, and basically we'll be able to get all of these exciting results eventually. And hopefully March next year.
Good question. Really good question. This is really a great question, especially with the work that you do to promote STEM and STEM for females. So Rita is asking for a bit of career guidance, if you would. She's an undergraduate in biological sciences and wants to pursue a career in astrobiology. She's wondering any ideas of maybe what to choose for a master's degree, or any maybe guidance with that, because you probably don't want to choose one for her, but what do you think?
Yeah, definitely. I would say one of the main things with a field like astrobiology is that because it's so multidisciplinary, there isn't really one route into it. I think as long as with what you're studying, you're also passionate about being able to link that to astrobiology and kind of further along that route, it wouldn't matter what kind of subject you end up taking at a master's level. I myself actually didn't do a master's. I went from an undergraduate in astronomy. I took a year working in a science centre doing planetarium shows about astrobiology, and then went into a PhD. And I know people that have come from zoology, geology, physics, engineering, and all ended up in astrobiology. The main thing I suggest is just if you know someone who's at a university that you think you might be interested in going to, just drop them an email, because people in academia are more than happy to help and give them with this kind of thing. So yeah, definitely contact people and see what they have to offer further down the line. But best of luck with it. And yeah, I look forward to working with you in the astrobiology field.
Good advice, really good advice. Yeah, and a good question. And I love that you are, supporting them and just making direct contact. Yeah, with someone who maybe they,
Yeah.
future mentor advisor, maybe. Okay, Surin Welch asks, are there risks of a contamination when we send missions to moons or planets with Earth-based organisms or organic materials that may be carried on the equipment? So this is also part of planetary protection. And why it takes so long maybe to get these missions launched and all packed up and ready to go. Go ahead.
Definitely. Yes, that is definitely a huge risk for these kind of things. And spacecraft have to be thoroughly sterilized before anything can be sent to another planet. Because like Lucinda said, planetary protection, we can't end up taking something from the Earth to another planet. So yeah, everything has to be thoroughly looked at to make sure there is no chance of any kind of contamination there. So the only other place where that isn't the case is the moon. So there isn't planetary protection on the moon, which is why when the spacecraft tardigrades crashed into it, it wasn't that much of an issue. But anywhere else like Mars or the icy moons, we would really have to consider that, because we don't want to end up contaminating that planet with something from the Earth.
So that kind of segues into this next question from Eddie Carpenter, who asks, what might happen to the organisms we've left on the moon, or if any by chance got to Mars from our rover missions?
So in terms of the tardigrades on the moon, again, they need that reanimation, which they probably wouldn't have been able to access. So nothing has happened with those being on the surface of the moon. The idea of things traveling between planetary bodies is an interesting one, because there is the idea that like life on Mars could have traveled between its moons, and life could have either originated on its moons and traveled to Mars, or vice versa. But it's not something I'm the biggest expert on, I'm afraid. But it's really interesting to think about. But I think generally things would tend to stay contained, unless linking on to the previous question about the meteorites, if something could travel via that.
Okay, another question, a question about micrometeorites. So, we have comet Neowise in our sky, and often have meteor showers from former comets who've come through our orbit. They're asking about these micrometeorites that come down every day on Earth. Might some of them have microbes?
So it's kind of similar to the other question about meteorites. And again, it would definitely depend on, because obviously when they land, they would burn up, and that would be an intense heat for anything to be able to survive, and also the impact, the pressure of that. So for anything to survive, it would have to be extremely tolerant to these things. So in my opinion, it's highly unlikely that something would be able to survive on that. But there is, again, the way that we can analyze these meteorites to learn more about the planets is really exciting themselves. So even if there isn't life on these meteorites, the fact that we can analyze them to learn more about these planets is really interesting.
Yeah, it is very interesting. I mean, more research to be done. Here's a really interesting question. Mazin Nasrallah asks, transient brines appear to be present at the surface of Mars at 205 Kelvin. That's way lower than anything known on Earth. And it seems if there's extant life on Mars, it will be on the surface. Do you know how deep we would have to go? Maybe they meant that it would be in the surface. So, so how deep would we have to go?
Yeah, so again, even if there was liquid on the surface, we'd have to be going far enough back that there was a thick enough atmosphere to stop all of this harsh radiation from coming through, because that's a big factor in whether anything could survive on the surface of Mars. In terms of how deep, I imagine if you're talking about if that was been buried, kind of how deep it was to go. I don't know exactly how deep, but hopefully with things like the upcoming ExoMars mission, which is going to have a drill to dig beneath the surface, we'd be able to find out a bit more about that. And so basically what is beneath the surface of Mars, because so far we've not really been able to probe under the surface. So hopefully in a few years' time, that's something that we'll know a bit better.
Yeah, so if all goes well, fingers crossed, and Perseverance gets launched later this month, or perhaps early August. It'll land, I think, in February of 2021. It always takes time to start getting data back, and NASA's been pretty good at making sure we get the data pretty quickly these days. Of course, after all the scientists have looked at it. But they're landing in this Jezero crater, which is, ancient impact craters, which has exposed the subsurface rocks, that we don't normally get to sample. So that's what's so exciting. And it's in this ancient lake bed. So if there was any life when there was liquid water on the surface of Mars, if there ever was, life would have then trickled down and perhaps been caught in any of the tiny sediments down at the bottom shale and what have you. So that would be really great, which is why I would be so excited to hear about the mission that's going to be returning the samples sooner than later. So really excited for all the missions going on. It's amazing that we're having so many missions to Mars this year, or so many countries as well interested in doing it.
Yeah.
Okay, your next question is from Chris Lee. And he asks, what was the final position on the Ellen Hills meteorite from Mars? Was it a microbe or not?
I think the final decision was that it wasn't. It was something that I think for a lot of people, there were structures inside a meteorite that came from Mars that a lot of people thought looked like they may have come from life. I believe the general consensus is that it just happened. There were these structures inside and that it wasn't from any kind of life. Yeah.
Yeah, I think you got it pretty much spot on that, it's not. And that's why we have to still do more experiments, more research. And that's exactly it. And that's what science experimentation is, you get your hypothesis, yes or no. And if not, then keep looking or keep experimenting. So excellent. So our Deputy Executive Director of the Royal Astronomical Society, Dr. Robert Massey, says, great talk, Rosie, and his 10-year-old daughter, Ada, likes it too. Hi, Ada. And he also mentions that Jane Greaves on the RAS Supermassive podcast is happening. So, and you talk about your podcast. You were also interviewed on the RAS Supermassive podcast. Is that what you have here on your last slide?
So that it was just Professor Jane Greaves that was on the podcast. So I haven't done a podcast for that, but I just hoped that the things that I've done have kind of linked in with what she's talking about there.
Sounds like we need to get you on one then. This has been a fantastic, and Professor Jane Greaves, I've interviewed her too. So, well, thank you to you for helping get the word out, and Dr. Massey for getting us the link, which is podfollow.com/supermassive if you want to tune into that. And so I guess he's giving us the link to your last slide. Okay, let's see. Here we go. We have about 2 more minutes, and 1, 2, 3, 4, lots of questions still. So I should have asked you, we could go a little bit over time, but it's completely up to you. If you have another meeting to go to or anything, I can understand, but maybe we can get through a few more. How do you feel?
Yep, a few more is fine, yeah.
Okay, let's see. Well, I'm not sure maybe you could help me here. Someone asks, could there be parasites in space? Maybe they're just using that as another term for microbes?
Maybe, I'm not too sure on that one either.
So parasites are yeah. So I guess we would use another term for them, because we don't really get to, if we haven't found anything in space, is that fair to say? And if we haven't had an opportunity to study them, to know their behaviors, I suppose, is that fair to say?
I would agree, yeah.
Okay. All right. Let's see. Jim Slight wants you to know that great talk. And you have a great future in front of you. Thank you, Jim. Very kind of you. Let's see. Carolyn Newberg also says, exciting talk. She wants to know, which A-level would you encourage students to follow to reach reach an astrobiology job or career pathway? All three sciences, which is typically what triple science, biology, physics, chemistry. And which degree at university might you recommend? And she says, thank you.
I think, again, it kind of stems back to the question of, as long as you're interested in the subject, it doesn't matter so much what you study. I think having an A-level in biology would help in the understanding of the kinds of life, but it depends what kind of route you wanted to take into astrobiology. So again, you could follow a biology route. You could go into astrochemistry, which again, you're looking at the kind of chemistry we might find on other planets. There's the geology route, so you could do geology at A-level and look at planetary science. Physics, if you wanted to go into more observations and exoplanets and looking at their atmospheres. So I wouldn't say there's one A-level that you would kind of need to go into astrobiology. I think it's more looking at what kind of side of it you're interested in, and then maybe following that. And kind of the same at university. It's the same kind of subject ideas, just what kind of bit of astrobiology you're interested in, because it's a huge subject area. To think about what parts you're interested in. And yeah, best of luck with your A-levels and deciding on university programs.
Yeah, good question. I think usually for higher tier, you have to take triple sciences anyway, if you're on the science track, typically. So you would have to do all three sciences. In my geology degree, I remember, and this is the United States, I had to take biology, chemistry, and physics concurrent, with calculus. So I had to do all three sciences at university anyway. I don't know if it's different here. I don't know the UK route for geology, but I'm sure geophysics covers a lot of the physical part of the science, obviously. Some of the people I've worked with prior who had degrees in astronomy then went into astrobiology. So you're absolutely right. You could really go towards astrobiology and specialize as you move up in your, your learning path from undergrad to PhD. So specialize, absolutely. They kind of all, friends who are get into chemistry, become geochemists, you never stop learning or never stop, trying to understand more of the bigger picture. And sometimes that means you need to take a course on something different than you got your PhD in. And it just really helps you kind of make all those connections. And of course, collaborating with other scientists too. Let's see, David Gwynn says, thank you, Rosie, and to the RAS, thank you so much, David. And he says, wonderful, and it has filled gaps in his knowledge. And he says, good luck with your studies. And I know you're almost there. I wanted so much to call you Dr. Rosie Cane on this talk. I know you're going to get that. You're almost, I'm so sorry to mention that, but.
No, thank you.
All right. Let's see here. Gemma Dank says, could you comment on the conditions thought to be required for the origin of life and how they might differ to the conditions required for sustaining microbial life?
Oh, so again, with astrobiology being a big topic, that is one huge field, is looking more towards the origin of life. So it's not something that I'm that versed in. I don't know that much about the conditions. We think that they would have happened, it would have happened aquatically. But other than that, sorry, there's not much more I can comment on that one. Yeah, my undergraduate degree was in astronomy, then I've learned biology through my PhD. So there's only so much about astrobiology that I can specialize upon.
Yeah, I think I kind of understand, I think what you mean, and maybe it's because like one of the expeditions I went on, we're looking at, I guess, microbial life, looking at 3.5 billion year old stromatolites that are fossilized up in Australia. Yeah, I guess you'd have to focus on biology, pure biology, maybe, to look at sustaining microbial life. And there's all different types of ways, I guess, for them to, environments to live in. I think that's kind of what extremophilic means. There's all different types of extreme environments that microbial life seem to be living quite happily in, which is just so amazing. So maybe that's for another talk to kind of go into that. There's a great questions, and they're also helping me realize maybe what's our next speaker to maybe talk about. So thank you so much, Gemma. Dr. Massey on again asking, do you think it's more likely that we'll find simple life in the solar system first, or whether we'll see evidence for it in the spectra of atmospheres of exoplanets?
Ooh, that's an interesting question. It's so hard to know. Like, it's anyone's guess which we'll end up finding first. We've got all these exciting missions that are hopefully launching soon to Mars and in flybys of Europa, and further out into the solar system. But again, we're advancing, learning more about the like the spectroscopy and ways of detecting the different things present in the atmospheres of exoplanets all the time. So if James Webb does launch next year, then there's a good possibility we might end up finding something that indicates like a biosignature, something like the presence of oxygen on a atmosphere of an exoplanet first. But we're kind of waiting on which spacecrafts are going to launch first currently. So yeah, it's a really good question, but not one I can give a definite answer to.
I think, yeah, he's just looking for what your hunch is about future. Yeah, good one. So listen, I think since we're 5 minutes afterwards, you probably need a break and some lunch and resting up for your next talk at 6 o'clock. So what I'm going to do is I'm going to make sure I copy the questions that we have waiting. And if you're okay with this, Rosie, I can give them to you later, and you can answer them offline, and then we can post them on our website. Will that work out okay for you?
Yeah,
okay,
that's absolutely fine.
So we're just going to say goodbye and thank you all for joining us for our 1 p.m. bicentenary lecture. We're going to be on again at 6 o'clock with Rosie Cane for the same talk. And I think you still can register if you want to listen to it a second time. Of course, this has also been live streamed on YouTube, so you're more than welcome to rewatch it again. Wanted to just also, so Rosie, thank you so much for being with us. And thank you again for helping us celebrate 200 years with the Royal Astronomical Society. And if you need to go, that is great, and we can see you later. But I wanted to also remind attendees here, as you're filing out, that next Friday, the 17th, we're also going to be live streaming the transit of Ganymede across the face of Jupiter directly from Mauna Kea, Hawaii, which is just going to be amazing. So we're going to have a full day from 11 to 4 p.m. of this observation. Actually, it's going to be an experiment that you could be a part of. I mean, that you're going to be watching actual scientists do, and they're going to be explaining the science and how they're doing it, what they're doing it, and what they see. We're also going to have a guest speaker, Professor Emma Bunce, who is going to be talking about, just like you mentioned, Rosie, Jupiter. She'll be talking about Jupiter and possibly the JUICE mission and maybe some of the things that we found with Juno. And so it's going to be an exciting day. Oh, also our RAS librarian, Dr. Sean Prosser, will be sharing with our attendees some of the collections that we have on early observations of Jupiter illustrations, some really beautiful, beautiful artifacts. So we hope you join us. That's going to be on the 17th of July, next Friday. So until then, we'll say goodbye. Thank you so much for joining us.
Thank you.